Problem solving can be so much easier when you know what not to do. Hi biology friends, it's Sarah, your other biology professor, and let's solve some problems with Chargaff's rules. Skip ahead to those problems if that's all you're looking for. Otherwise, we're going to get started with a little bit of review on nucleotides and the rules. Now, by the 1940s, we already knew stuff about DNA. Like, we knew that chromosomes were made up of protein and DNA. And we knew that DNA consists of nucleotides, which are made up of a phosphate group, a 5-carbon sugar, which in DNA is deoxyribose, and a nitrogenous base. And the base is what changes. That's what makes the different types of DNA nucleotides. There are 2 categories of bases. The purines, which consist of a double ring structure, and those are going to be A, adenine, and G, guanine. Pure as gold. The purines are A and G. And you've got the pyrimidines, which consist of a single ring structure. There you've got cytosine, C, thymine, T, pyramid, think sharp, cut. The pyrimidines are C and T. One thing we didn't know was what happens when you put those nucleotides together. What was the structure of DNA? Now, one popular idea at the time was called the tetranucleotide hypothesis, which suggested that you take one of each nucleotide, combine them to form this tetramer, and DNA is made up of repeating subunits of that tetramer. Now, that meant that the nucleotide bases are going to be present in equal amounts in a DNA molecule, and it also meant that there wasn't any variation in the DNA molecule. They're all kind of the same. After Avery, McLeod, and McCarty published their work showing that DNA could transform bacteria from one type to another, suggesting that genes are made of DNA, not protein, the biochemist Erwin Chargaff was like, somebody should look into this. If DNA is the molecule of inheritance, then there should be differences between the DNA of different species. There should be variation. So Chargaff got DNA from different species. He used acid to pop the nucleotides apart from one another, paper chromatography to separate them out, and then finally UV spectrophotometry in order to quantify how much of each nucleotide base was present in a given sample. He found that for a particular sample, the amount of G was roughly equal to the amount of C. The ratio of G to C was pretty close to 1. And similarly, the amount of A was equal to the amount of T. Now, the reason for this, complementary base pairing, would be solved later and would be super helpful for Watson and Crick and their double helix model of DNA. Another important finding from Chargaff was that DNA does in fact vary between species. Now, this helped to falsify that tetranucleotide hypothesis and give more weight to the idea of DNA as the genetic material. But it's really that first rule that you got to keep in mind for solving most of these problems. A fairly typical problem looks something like this. If the DNA of a certain species is 35% thymine, what percent is guanine? Well, if T is 35%, then that means what else is 35%? A, right? Because A equals T. So T is 35%, A is 35%, add those together, they make up 70%. So 30% is what's left. And this is the number one mistake that I see students make. You see that 30%, you're tired, you rush, you pick 30% as the answer and you move on, but no. There's one more tiny step. Remember, that 30% represents the amount of guanine and cytosine, right? G equals C. And those together are 30%. That means that C is 15% and G is 15%. Let's do another one and we'll ugly up the numbers a little bit here. So a molecule of DNA is 31% guanine. What are the percentages of all 4 bases? Well, if we're 31% G, we're also 31% C, right? G equals C. So G is 31, C is 31, add those together, G plus C is 62%. That means we've got 38% left for A and T. A equals T, and together they're 38%, so that means A is 19%, and T is also 19%. Now here, again, we got ugly numbers. So if you've got 2 seconds, go ahead, toss them in and make sure they add up to 100%. A plus T plus G plus C, just to check your math. Just in case somebody tries to get a little bit sneaky with you, there are exceptions to the rule. Chargaff didn't know this, but we do. This rule is based on complementary base pairing, which means it does not work with single-stranded genomes. In fact, Chargaff actually looked at a bacteriophage, a type of virus, that has a single-stranded DNA genome. And there he found that the ratio of A to T was 0.77, which is not even close to the 1 that you'd expect. So when you're tackling these problems, keep in mind A equals T, G equals C. Try not to make silly math mistakes. That's the hardest part, and you'll be just fine. In the meantime, if this was helpful, drop me a like so that other students can find it as well, and subscribe for even more biology, content, problem solving, and tips from me. And I will see you next time. Bye.